to one and all present here. Myself Reshma, doing PhD under the guidance of Dr. V. Mary Hensa from the Department of Botany in ST Hindu College, Nagarkoil. Today I am going to present the topic on phytochemical analysis of Lepidognathis barbary Campbell. Introduction. From the emergence of humans on the earth, human depends on plant as a source of food and therapeutic aids. Each and every part of medicinal herbs are used to treat diseases. Phytochemicals are the natural biotic compounds found in plants such as fruits, vegetables, flowers, leaves, and roots that works to act as a defense system against the disease. Plant products have been a part of phytomedicine since time immemorial. This can be derived from barks, leaves, flowers, roots, fruits, and seeds. Aim of my work was to study the taxonomical characters of the experimental plant Lepidognathis barbari and to analyze the physicochemical properties of the soil where the plant is grown to find out the quantitative and qualitative analysis of the plant. I have collected the plant from the GVN College campus, Koilpatti, and this plant was identified and authenticated by BSI Coimbatore with the accession number 651. A voucher specimen was deposited in the herbarium of EST Hindu College Nagarkovil, and the plant was shade dried, powdered, and stored for further analysis. Thus, soil sample from three different places of the plant grown is collected at a depth of 1 to 10 centimeter, and it is combined to form a homogeneous composite sample. The soil sample was air dried for 7 days and it is grinded in a clean mortar and pestle, and it is sealed and stored for further analysis. So this table is showing the method used for the estimation of the parameters. Qualitative analysis of the phytochemicals. So this way the procedure that is used to find the qualitative analysis of the phytochemicals. First, detection of carboxylic acid and tannins, steroids, detection of flavonoids, detection of glycoids, detection of proteins by Bradford method, detection of phenol by ferrichloride method, saponin test, the test for alkaloid smear test, saponification test, gum test, detection of flavonoglycosides, detection of carbohydrates, detection of resins, and tests. Quantitative analysis of phytochemicals. For lipid estimation, vanillin method is used. And for total tannin, a fall-in cocktail method is used. And for estimation of reducing sugar, Benedict test is used. For protein estimation, Bradford method is used. For phenol estimation, falling cocktail method is used. For vitamin C, it is analyzed by spectrometer with 540 nm. For total flavonoid content, it is measured by calorimetry. Moisture content. The sample of 1 gram was transferred into the beaker and the beaker was closed with the lid and kept in a hot air oven for 500 Celsius for 4 hours. Then it was cooled in a desiccator and weighed. The loss in weight represented the moisture content of the sample. Total ash. About 1 gram of test samples was kept in an insulated dish and heated for at least 13 minutes in a muffled furnace for 550 degrees Celsius, cooled in a desiccator and weighed to the nearest 0.01 gram. Wood fiber method, muslin cloth method is used. Results. This is the morphological studies of the Lepidognathis barbari. It is rare, erect, small undershrub which grows from 30 to 70 centimeter height. The stems are well branched, four angle, pubescent and green to purplish when young and ash colored when old. Cylindrical, branches wide, leaves sessile, isophilous, opposite desiccate, oblong to narrowly elliptic, and inflorescence is spike with converged factorial bracts which are alternately arranged on the axil of the leaf. Bracts are sterile. Bracts 4 arranged in two pairs, heteromorphic, lands ovate to ovate, and it grows on dry, sandy, gravelly terrain of wasteland. It is distributed in areas which receives less rainfall and remain dry for a large part of the year. It is distributed in various parts such as Coimbatore, Erode, Theni, Dindigul, Tirunelveli, and Virudhunagar. So this has some medical properties and it is used in the treatment of fever, psoriasis, epilepsy, and other skin disease. It also shows some pharmacological important activities such as antipyretic, antiurolytic, anti-inflammatory, hypoglycemic, wound healing, and immunosuppressive, and boosting fertility. This is the table showing the physical chemical properties of the soil sample collected from the area. And this is the table that showing the phytochemical compounds. So this shows the presence of resin, carboxylic acid, flavonoid, carbohydrates, saponification, protein, saponins, and gum. table shows the lipid estimation. The total content of the lipid was 1.7, and the total tannin was about 81.0 mg, and the presence of sugar shows us is the total content of 30.1 mg, and the protein content present in the sample was 30.8 mg, and the total phenol content was about 24.3 mg, and the flavonoid content was about 30.1 mg, and the vitamin C was 1.2 mg, and the total ash was about 0.9. Conclusion: the current investigation described the morphology, physicochemical parameters, and qualitative and quantitative analysis of the phytochemical analysis. And the physicochemical properties of the soil shows the color, the texture, pH, temperature, water holding capacity, moisture, bulk density, electric conductivity, organic compound, nitrogen, phosphorus, potassium, calcium, magnesium. So the phytochemical analysis showed the presence of resin, carboxylic acid, flavonoids, saponification, proteins, saponin, gums using isopropanol solvent. Qualitative analysis shows the presence of lipid, tannin, sugar, protein, phenol, vitamin C, total flavonoid, moisture, total ash and crude. So this work confirms the importance of plant as a powerful source of diverse bioactive pharmaceutical metabolites that are a great importance for the development of new drugs. That's all. I conclude my presentation. Thank you all.